All right. So there we go. Okay, so Dr. Foshay is the Senior Director of Genetic Therapy Programs for the Cystic Fibrosis Foundation. She was trained as a cell biologist and completed her doctoral and postdoctoral studies at Georgetown University and the University of Chicago. She's very smart. Her first experience with patient-centric research was at Inova Health System in Northern Virginia, where she led the brain tumor research lab for 4 years and worked to develop personalized chemo screening platforms for glioma patients. The passion that she took away from this patient community is what sparked her to come to the Cystic Fibrosis Foundation. She brings a unique perspective and expertise to her current role, and she leads the industry-based Path to a Cure portfolio and the preclinical development subgroup of the Genetic Therapy Working Group. So that's a big group. Kara's also the mom of 2, an avid runner, a home chef, and an advocate for women and girls in science. So I want to welcome Dr. Foshay and thank her for taking the time tonight to be with everybody. And I will close my camera off. See you in a minute.
Thank you, Margie. That was a great introduction. And thank you all for having me tonight. I'm really excited to do this. This is actually my first chapter science update. So welcome your feedback at the end and looking forward to all of your questions. With that, I will share my screen. This one. Okay, and can you all see my full screen slides?
I can, yes, Tara.
Yeah.
Okay, thanks. Great, so we'll get started. I'm calling our update tonight, Navigating the Path, Approaches and Progress Toward Genetic Therapies for CF. And I did see on the attendee list a few names that I know, and some of this might be familiar, but hoping it's the update you were all looking for. So today's roadmap will cover a little bit of Bio 101 to level the playing field. We'll talk a bit about the unmet need and why genetic therapies are the answer. I'll go over some of the scientific challenges that are specific to CF, and then introduce you to our current Path to a Cure portfolio. I'll follow up by what's next, and that'll be a little bit more than just science, but some of the other issues we're facing with regards to regulatory and clinical trials in the gene therapy space. So starting out with Bio 101, it's been a while for me, but this is what we call the central dogma. So DNA, which contains your genes, is transcribed into mRNA. mRNA is a single-stranded copy of the information that was in your DNA. mRNA is translated into protein, and protein are the functional building blocks that make your cells work. When we talk about CFTR, we talk about a protein that functions as a chloride transporter, and I'm sure that's familiar to most of you. Now, DNA contains genes, and genes are made up of exons that contain coding sequence or the important information for making a protein. They also contain introns or non-coding sequence. When DNA is copied into mRNA, the entire gene is copied. But then the introns are removed through a process called splicing. So these are actually cut out, the non-coding information is removed, and you have a mature mRNA. This mature mRNA is then translated into a protein. And I think on the side here, it's interesting to note that the introns actually make up a lot of the gene. So the size of CFTR before and after splicing is quite a bit different, about 190,000 nucleotides versus 4,440. So why do different mutations have different outcomes? Well, that's because each exon encodes a different part of the protein. So depending on where your mutation is located, it may have a bigger effect on protein function than others. And that's because some part of a protein are more important for function. So for instance, how does F508del impact CFTR function? Well, F508del is a mutation that causes CFTR to misfold. Meanwhile, G551D, another common mutation, causes CFTR to not open and close properly and doesn't allow chloride transport. So in the end, neither allows chloride transport, but it's for a different reason. Modulators work by correcting protein function. In the case of F508del, correctors can help the protein fold properly and translocate to the cell membrane where it needs to function. In the case of G551D, potentiators can help the chloride channel open and close so chloride can flow properly. But my point here is that modulators act on protein and they don't correct mutations in your DNA. And that takes us to the unmet need. Patients who have mutations that result in a total lack of protein. When we talk about patients that don't have CFTR protein at all, we think mostly about nonsense mutations. But I'll add that there are other mutations, such as large deletions, that also result in a loss of protein. So what's a nonsense mutation? A nonsense mutation introduces a genetic stop sign. So in the process of translation, the ribosome traces along the mRNA, it encounters a stop sign and it stops making a protein. CFTR modulators aren't effective because there is no protein to act upon. I thought I would add in here because we talk about nonsense mutations and read-through. And read-through agents are not necessarily a genetic therapy, but they're something of interest to a lot of patients. So I thought I would add this in, that nonsense mutations cause a degradation of mRNA. So we talked about splicing. Here's an example of a nonsense mutation, W1282X. It's in red in exon 23. When splicing occurs, you have your mature mRNA and the genetic stop in red. Read-through agents can encourage the ribosome to skip over that stop sign and make a mutant protein. So that's one therapeutic option. We also talk about something called nonsense-mediated decay inhibitors. When a cell encounters a mutated mRNA, it degrades it because it thinks it's not the correct sequence. If we block that process of degradation, we can restore mRNA function. In combination with a read-through agent, we could potentially restore expression of a mutated but functional protein. So how about genetic therapies? Because I think a lot of us are interested in therapies that don't just help nonsense mutations and don't just help specific mutations, but could help anyone with CF. So I'm going to borrow a metaphor from my good friend Hilary Valley at the CFF lab. She presented these in the plenary at NACFC last year, and they're tweaked a little bit for tonight. But if CFTR were a car, you could think of car problems as CFTR mutations. And so here you've got flat tires. And there are a few flavors of genetic therapies for CF. And you can think about them in the ways you can get around your flat tires. So the first genetic therapy we'll talk about is antisense oligos. These are applicable to nonsense mutations, just like the NMD inhibitors and read-through agents I just spoke about. But antisense oligos act as sort of a patch. I show it here as bike tires. They're not ideal, but they might work. mRNA therapy is sort of like a rental car. It gives you something to drive for a short amount of time. mRNA is very short-lived in the cell, so this is not a permanent therapy. In fact, it would probably have to be redosed really often. The next category is gene replacement, and that's kind of like getting a brand new car. That's introduction of an entirely new copy of the CFTR gene into your cells. Now, that copy of CFTR is yours for the life of the cell or in the life of the car. Because gene replacement does not integrate into your genome, it doesn't last forever. It's not a permanent therapy. Then finally, we have gene editing. Gene editing is a permanent change to your DNA. It's sort of like repairing the car you had so you can keep driving it. Antisense oligos, so we'll start here. As I mentioned, nonsense mutations are like a genetic stop sign. You've got your RNA, you've got your mutation. Antisense oligos are like a mask. They cover up that stop sign so that the cell doesn't see it. When splicing occurs, it skips that mutated exon. So you're missing exon 23, and the result is a shortened but still functional protein. So back to our car analogy, it's like patching the tires or using a bike tire. It's not exactly right, but it works, and that's what's really important. When we think about what an antisense oligotherapy would look like if it was a drug, when it becomes a drug, it would target the differentiated lung epithelial cells that are replaced by new cells over time. And the actual therapy would be these little pieces of RNA that block splicing and allow for production of a shortened but functional protein. Because of those factors, the duration of this therapy would be short, probably a couple days, and would require frequent redosing. This would be short-lived restoration of CFTR function, but only for people with specific nonsense mutations. This is not a universal therapy. The next three approaches I'll talk about are universal. Well, the next two are universal. mRNA therapy, it's like a car rental, a short-term fix. It would target, again, the differentiated cells of your lung epithelium that are replaced over time. The actual therapy would be non-mutated copies of CFTR mRNA. They would be translated in your cells into CFTR protein just like any other mRNA. Again, mRNA is really unstable, and so it only lasts for a few days, and dosing would have to be frequent. The outcome, again, would be short-lived restoration of CFTR protein function, but this would be for people with any mutation, because the mRNA is the entire CFTR mRNA. Now, gene replacement therapy is a little bit different. It would still target the lung epithelium, but it could also potentially target basal stem cells. So those are the cells that repopulate the lung epithelium. The therapy would be a non-mutated copy of CFTR DNA that would be inside your cells in what we often refer to as an episomal plasmid. So it's not part of your chromosomes or your DNA, but it's kind of separate on the side. It hangs out and is lost over time due to cell division, but will last a lot longer than mRNA. So we know a DNA gene replacement therapy would need to be redosed, but the timing isn't as clear. It could probably be anywhere from months to years, depending on how many cells took it up and how quickly they replicate. Again, this is a transient restoration of CFTR protein function, and it would be amenable to people with any mutation. And finally, there's gene or DNA editing therapies, and I'm sure you've all heard about CRISPR in the news. It's pretty exciting and pretty cool. This therapy would look a little bit different than the others I just described. When we think about editing or making a permanent change in your DNA, we really want to target the stem cells that will be there for the life of a person and will continuously repopulate the cells of the body. There are stem cells in the lung, and they're called basal stem cells. These are the cells we would want to target for gene editing. Every time they divide to make a new lung epithelial cell, those cells would contain the edit. The therapy here isn't RNA or DNA. It's actually proteins that edit DNA. They would target CFTR and a person's specific mutations for correction. Again, the edit would last a lifetime. It would be passed to daughter cells, and it would lead to permanent restoration of CFTR protein function. Now, each editing therapy that would be developed would in general be specific for a mutation, but the technique in general and the approach in general could be applied to any mutation. Now, This sounds good, but when we think about drug development and the regulatory process, developing 200, 300 different drugs to edit all the different mutations that cause CF would take a long time, cost a lot of money, and would be really inefficient. So when we at the CF Foundation think about the shoot for the moonshot approach, we think about what we call super exons, one strategy to repair most mutations. Now, I talked to you a little bit about exons, right? They're the coding part of the DNA that is present in the mRNA and is still present after splicing. So we know that mutations that cause CF span the entire gene, all 27 exons. But if we made what we call a super exon, a piece of DNA that contained the coding information from exons 2 through 27, and inserted it into the endogenous CFTR gene, we could repair most mutations with one single therapy. Now, there are a couple academic labs and companies pursuing these approaches, but I'm going to tell you that these are far removed from the clinic right now. These are still very experimental, but really exciting approaches. And it's something we're working really diligently toward. So I'm going to highlight a few success cases, examples of genetic therapies in other diseases, because I think it makes it a little more real. There are no FDA-approved gene editing therapies right now, but there are some that are moving forward. And I'll highlight this one, NTLA 2001. It's a product developed by Intellia Therapeutics to treat ATTR amyloidosis. And this editing approach is in clinical trials right now. It's targeting the liver, which most scientists would tell you is probably the easiest place to target. But initial results are promising. So even though I'm saying editing therapies are far in the future, they are making progress, and it's really exciting to see. Now, for the other types of therapies I described, there are FDA-approved therapies. So ASO exon skipping, there's a drug called Exondys 51 for Duchenne muscular dystrophy. It's a weekly IV infusion and results in a truncated but functional protein, just like I described would happen for CFTR and people with CF. But there are some differences. DMD has a very long half-life and is expressed in non-dividing cells. That's different from the dividing epithelium of the lung. For mRNA therapies, we have COVID vaccines. I think this has done a lot to push the field of mRNA therapeutics forward. So COVID vaccines, as probably most of you know, consist of 2 intramuscular injections and boosters as indicated. These target your immune cell antigen recognition pathways, so that's a different target than CF. And a functional protein is not required for efficacy of a vaccine. You need a piece of a protein to be recognized by your immune system, but you don't need to make a functional ion transporter. So the bar is a little bit higher for CF. And finally, for gene replacement, I'll highlight Zolgensma for spinal muscle atrophy. This is a single IV infusion of AAV9, so that's an adeno-associated virus, carrying the gene SMN1. This targets non-dividing motor neurons. So again, a non-dividing target. And SMA is a disease that is invariably fatal by age 2 without any intervention and no other therapies to improve outcome. And so the disease severity here increases a tolerance to risk when it comes to experimental therapies. And I'll just highlight those differences here that set the bar a little bit higher when we think about gene therapy for CF. I'll dive a little deeper into some of those scientific challenges now. Are there any questions, by the way? I'll just pause for a minute. No? All right. So the first challenge I'll highlight is size and composition. So I think this is just sort of interesting to think about. We see on the left a picture of the molecular structure of ivacaftor, which has been published. And this is a small molecule. Small molecules easily diffuse into cells. They go throughout the whole body. On the right is a picture of 4 RNA nucleotides. Now, I mentioned earlier in this talk that full-length CFTR mRNA is 4,440. Here it says 6,128 because what I didn't tell you is there are untranslated regions. There's other bits on the ends. So the entire full-length human CFTR mRNA is about 6,000 nucleotides, and this picture shows 4. So these are big, bulky things that we have to get into the cell, and they do not diffuse in just like a small molecule. Instead, they require a delivery vehicle or a way to package them. Think about when you have to send your Amazon package back, right? You have to repackage it, seal it up, and get it to where it needs to go. Although that's not a good example anymore because now you don't have to package, right? You just take it to Whole Foods. Anyway, gene therapies require delivery vehicles. And I highlight here the 2 most common types. One are viral vectors. So we can repurpose viruses to express genes we want instead of viral genes. In the case of a CFTR directed therapy, CFTR DNA would replace the viral genome. The most commonly used viral vector for gene therapy is adeno-associated virus, and it's been widely tried for many diseases. It does not change your genome, but it has a pretty small size. So the geometry of a virus is very consistent, and it can only fit a certain amount of genetic material. CFTR, being a very big gene, barely fits. We also know from previous studies with AAV that redosing may be a challenge with AAVs. These are viruses that you would see or encounter naturally in life, so some people are already immune to them. We also know that our bodies rapidly form antibodies against AAV, and so if you have one dose, you may then be immune to any subsequent doses. The other most common viral delivery vehicle that we see or hear about, I think in the news, are nanoparticles. And there are a lot of flavors of nanoparticles. They can deliver a variety of cargos. And so the size is not a limitation. You could easily package CFTR or multiple copies of CFTR inside of a nanoparticle. They're pretty effective for redosing. You don't have that immunization or neutralizing antibody problem that I described with AAV. Although we are seeing more and more that there is a potential for an inflammatory response over multiple doses. And so we think about those side effects from your COVID vaccines. These are not totally benign, and we're working to, figure out what's causing that. I think the thing about nanoparticles is that targeting specific cells is really challenging. Viruses over time have gotten really good at targeting the cells that they want to infect, and nanoparticles aren't quite as efficient. Another scientific challenge we're facing in CF is identifying the right target cell. So there are a lot of cells that express CFTR in your body. We're focusing here on the lung and that differentiated lung epithelium I mentioned. And we know that there are ciliated cells, ionocytes, secretory cells, basal stem cells, and from recent work done from the CFF lab and some stem cell consortium members, we know a little bit more about all these cell types. We know that the basal cell is a stem cell of the airway, and it repopulates ciliated cells, ionocytes, and secretory cells. But there's a lot of shading going on here, and that's because not all ciliated cells are the same, and not all secretory cells are the same. And we also know that the makeup of, or the distribution of these cell types within the epithelium changes as you go from the proximal to the distal lung. So there's a lot of cell types, and targeting the right one is a little bit complex. Here we show that the most probable target for a gene therapy that is transient is secretory cells and ionocytes. So those express the most CFTR. The most probable target for a gene editing or repair strategy is the basal stem cell that would repopulate the epithelium over time. The next scientific challenge is the hostile environment of the CF lung. So I don't think I need to tell you about the thick mucus that CF patients have, but it impairs clearance of the lung, and it's full of proteases that can degrade proteins, nucleases that can degrade gene therapies. You have immune cells that can recognize and destroy or take up your therapy. So there are a lot of cells called macrophages, and these are vacuum cleaners of a cell. They clean up all the debris and dirt that they find, and they love to gobble up a gene therapy. The other challenge, I think, is route of administration. Are you going to deliver this as an inhaled therapy or a systemic therapy? Because both have their challenges, and both make it hard to target specific cells of interest. Finally, CF is a multi-organ disease. I've talked a lot about the lung just now and the difficulty in targeting the lung, but the ultimate long-term goal is to fix CFTR in all cells that normally express it. And that's a lot of cells throughout the body. And so you can see, I think, that we have a lot of challenges to face in CF before we get a really good gene therapy to help all people with CF. And I think a lot of times we think about the major hurdle when we think about all of these, targeting the right cells, how to deliver it, how to get through the mucus. The biggest barrier right now is delivery of genetic therapies. With that in mind, I'm going to highlight some programs in our current portfolio. So we've separated our portfolio in the industry space into transient therapies that will restore CFTR, curative therapies, so those are editing type therapies, delivery vehicles, which I just explained, splice correction, so these are your antisense oligos and read-through programs that we talked about, and finally, safety and immunology. And that's a newer bucket for us, so maybe I'll start there. I highlighted some of the issues with AAV and development of antibodies and some of the inflammatory responses that could happen with LNPs. And so we're actually starting to invest in products and programs looking specifically to enhance the safety of gene therapy. This is a new area for us. That's why there's only one program highlighted there. There are others in the works, and we're really excited about what we're seeing in that space. So I'm going to move from here to programs that are in or near the clinic in the gene therapy space for CF. 4DMT is the first one, and that one is in clinical trials right now. That is an adeno-associated virus containing a delta R CFTR. So it's not the entire CFTR, but it's a shortened and functional version of it. That is in phase 1/2 and enrolling 21 patients in this phase. It's ongoing now, although we don't have any data from it to date. The next one I'll highlight is Crystal Bio. So this is another virus, but not AAV. This one is an HSV virus, and again, contains copies, this time of full-length CFTR. So this, I believe, has initiated in Australia. This is an Australian company, but is projected to start hopefully next year in the US. Arcturus is an mRNA company. They are developing an mRNA, CFTR mRNA, in a lipid nanoparticle, and they are projected to do a CTA filing in Europe toward the end of this year. Splicense is developing a read-through therapy. They are projected to start clinical trials near the end of this year or beginning of next year. Vertex is developing an mRNA LNP therapy. We don't know much about that, but we've heard from their news briefings that there's a projected IND filing toward the end of this year or next year. And then Recode, another mRNA program that should have an IND filing mid-2023. So pretty dense on the mRNA programs and pretty dense on the viral-derived gene therapy programs. The last one there that I didn't mention is Spirovant. They also have an AAV program. We know it's in late preclinical stages and expect it to come toward the clinic soon, but we don't have a concrete date there. So when we think about what's next, one of the focuses is diversifying our portfolio. So again, I highlighted 3 viral programs and I think 3 mRNA programs that we know are in or near the clinic. And so when we think about how to move toward the future, we want to diversify, right? We want backups in case these don't work. And so our portfolio expansion is really focused on delivery vehicles right now. So I highlight some other types of delivery vehicles here, happy to answer questions on them afterward. So there are other viruses, non-AAV, and virus-like proteins. So these are actually not viruses, but they look a lot like a virus and enter the cell a lot like a virus. And they're very, very new. Nucleic acid, so we've seen programs that are actually building big scaffolds and structures out of DNA that can enter the cell as a delivery vehicle themselves. Cells as a vehicle, can you package something in a cell and deliver it? Extracellular vesicles, so these are little bits of cell membrane that bud off and you can package them full of mRNA or DNA for uptake by the cells. And then I have nanoparticles here, and that is to highlight other non-lipid nanoparticles, so polymers and other chemicals. So it's a pretty diverse and vast space, and we are doing our best to landscape it and find the best technology. And the last thing I'll highlight here is the little gray box that says venture creation. We're so invested in delivery vehicles that we have a few venture creation programs that are focused on building CF delivery companies. So we are hopeful that we are going to find the next best thing for delivery. I think that portfolio expansion and the new types of companies we're looking at is leading us to think about really creative investments. So when we think about our history with modulators, we were investing in companies that knew and focused on CF. And that's not so much the case anymore. The companies that are building delivery vehicle technologies usually have a vast library of particles. So there are building blocks of a nanoparticle that can be combined into many, many different flavors of the same particle. And the picture on the right is just meant to highlight how many different parameters you can change in one nanoparticle. The way they come together as a package changes where they go inside the body. And so most delivery companies can serve many diseases. They're what we call a platform company. They might have a CF program. They probably have a liver delivery program. They probably have an eye delivery program. Those are sort of the low-hanging fruit. They may have a CNS program because there's a lot of interest in the central nervous system. But the point I'm trying to drive home is they aren't CF companies. And so we need to get creative in the way we invest and the way we manage alliances with them to make sure CF starts as a priority and remains a priority throughout the company's development cycle. And that's a new space for us. And I wanted to just highlight it here in this group so you know that it's all about science, but there are a lot of factors. And that's why I think, the path to a cure is really complex. And so I'll also highlight uncharted regulatory and trial requirements. With only a few FDA approved gene therapies, the entire drug development community is really still learning what the FDA requires for gene therapies. And I know this picture on the right is kind of tiny and it came from the FDA's website, but what I want to highlight is that not all genetic therapies have the same regulatory requirements, and that's because not all genetic therapies have the same risk. So we are doing a lot of cross-functional work between the science team, the clinical team, the TDN, and the policy and advocacy team to really understand the regulations and what they mean for people with CF. And this is going to help us determine what the ideal trial size might be, the ideal trial duration, and the ideal endpoints. I'll also highlight that for some types of therapies, we are looking at long-term follow-up that's 10 years and up, which is a really long time. And we're working to engage with the FDA and talk about what that means and whether patients would be able to enroll in multiple studies or a new study while they were still in a long-term follow-up for another one. And finally, there's a few people on the call here who I think are maybe better versed to talk about this than me, but education efforts. So I'll highlight the work of our gene therapy working group, which has a couple subgroups, but one of the major efforts is to educate clinicians, research teams, and patients about gene therapies and to give them a glossary of terms that are the same. So when a patient asks a question, the clinician understands the language they're using and can answer clearly. We're also working on the science side to standardize our guidance to sponsors, what type of data we want to see before you can enter a trial, and to develop guidance on how the trials should be designed, endpoints, what the informed consent should look like. And we're doing this with input from leaders in the UK as well. And so I'll just end with this last slide here, which is not live yet. So here's your sneak preview, but this is suggested questions for a patient to ask who is interested in enrolling in a gene therapy trial. And I think this is one of the coolest pieces of information that we've put out of the gene therapy working group. And I think it'll really help the community as we move forward in this space together. So I will end there. I'm sorry I talked a little bit longer than I intended to, but happy to answer your questions about the science, the regulatory issues, anything I can do. So thank you. I will stop sharing.
Hi, everyone. We don't have any messages in the chat at the moment, but if anyone would like to even come off mute and ask a question, I see it looks like there is a hand up. Oh, Dr. Germana, come off mute and say hello.
Can you come off mute?
I don't know. Is she allowed to, Chris?
Yep, I just gave her access to come off mute.
Okay, great.
There we go.
Great. I'll put my camera on. So Kara, I'm the CF Center Director at Cohen Children's, which is located right at the border of Queens and Long Island. So I and my colleagues in the New York area, we've been doing this for many of us over 20 years. So we've seen the old days and we're happy about these new days.
Yeah.
We're always thankful to the foundation because they share our passion in finding a cure to help all of our patients. I have a few thoughts. I thought your presentation was very thorough and easy to understand. So, you know, we as sort of the guinea pigs for you.
Thank you.
You know, I think you did a great job. I think you kept it interesting. So I appreciate that because it's sort of heavy material. You know, Sammy Galvin, who just is our research nurse coordinator, and she just said, you know, great job, Kara. She and we've been talking about like how we talk to our patients and explain this. And I use the car model because I'm a car person. So I use that all the time in CF. So I was really, I was so happy to see that. Because I think bringing this level of material down to the level where a lot of people can just understand it is a great idea. If there's a way to translate it into Spanish, that would be great because we have a very large Spanish population in this New York area. So if you could, that would be great. I think I'm sorry.
No, I'm just agreeing with you. Yeah, I think that's a great idea. So I know that the materials I mentioned aren't out yet. They are coming out soon, I think, on our website. I love the Spanish translation idea. And I think that the folks on that working group may be one step ahead. I know that they've held focus groups in Spanish to find out what type of information that community wants. I'm unclear whether we have them translated yet. But I can follow up and find out.
Yeah, and the other,
I thought Lynn might know. Yeah, many of the materials would be in Spanish. Lynn's involved in that subgroup.
Right.
So.
And my only other is, are we able to use some of your slides, because I have to teach this to colleagues, so I wanted to know if there be a way to borrow them or steal them shamelessly.
Yeah, I think that's fine. I don't think there's anything confidential on there, so it should be fine to reuse.
Yeah, usually Marjorie, I think, helps us with that.
Yep. I can send them to her and she can.
Great.
Yep. Well, thank you, Joan. Appreciate your comments. Thanks for joining tonight.
Great job.
And thank you, Lynn, for chiming in. Any other questions? We have one, I see. How many years are we looking at for gene therapy? So I think it depends. I think mRNA therapy will be here soon. We already have the AAV therapy in the clinic. I think the question is, are these therapies going to get to enough of the right cells? And that's the big question we don't know yet, because delivery is such a challenge and finding the right model to study it is really hard. And as good as any models might be, they aren't people. And so we want to see companies build strong packages and we want to have confidence going into them, but we all know that drug development is hard and doesn't always come through. So I think we're hopeful for the trials that we have coming. There were 6 I highlighted. But in terms of when they would actually be approved, I will tell you that the 4D molecular trial is moving very slowly because they're very concerned about doing it right, monitoring safety, and enrolling slowly. So I think we're talking about 5+ years before we would see an approved gene therapy if these initial ones worked. So still quite a ways. Yeah. Same with nonsense mutation. So I think splice sense is one, but it's for a specific mutation. I think I'm going to stick with my 5-year range for that. Is it more complicated with one nonsense gene and one rare gene? So that's a good question. We know that people that have one carriers of CF do pretty well, right? They might have, there's some literature that says they may have some mild symptoms, but if we could have everyone at that level, that would be great. So if you could, with a read-through therapy or an antisense oligo, restore that one nonsense allele to the level of a wild type, you'd be doing pretty well. So you only really need to correct one. Does that answer your question, Beth? But even in that case, with one nonsense or one rare, an mRNA therapy or a gene replacement therapy would work, because those are amenable to any mutation, and that's the beauty of those. Any other questions?
I
Yeah,
was
I
gonna say,
agree.
I'm gonna come off mute and ask you a question, Dr. Foshay.
Yeah, go ahead.
Why are there so many companies that are focused on lung-only genetic therapies?
Yeah, that's a really good question. And I think the main answer is delivery. So like I highlighted, delivery of gene therapy is not as easy as delivery of a small molecule that will diffuse through cells and target everything. So for every gene therapy, you have to target the correct cells. And it's hard to target everything. One thing we've seen with a lot of experimental therapies and things that have not moved forward is that it's really easy to target the liver. So that is why there are gene therapies for the liver. That is where nanoparticles want to go. That is where virus wants to go, because that's how your body normally clears things. If you want something that doesn't just go to the liver, the bar is a lot higher. And so companies are forced to pick an organ to start with. And because the primary morbidity and mortality of CF comes from lung disease, that's where they've chosen to start. It makes a lot of sense, but it's just a factor of how these things enter the cells. It's not as clear cut, or it's not as easy as a small molecule. I see one other question in the chat.
There's one in the Q&A from Dr. Germana. It says, did I understand correctly that it will take an additional 5 years for a medication for our patients with nonsense mutations?
So I don't have a great answer. There are no, I don't have a great answer. We have some shots on goal there. The progress is not totally clear. It could be faster for read-through therapies. So I was thinking about the splicens antisense oligo when I said that, but there are some read-through agents that are moving, and it's a matter of seeing how well those work. I think the issue is going to be getting them to be effective enough that we get enough CFTR to have an effect. So again, we need the equivalent of one good allele of function, and probably less than that. Studies say that we need about 20% of wild type function to get a good clinical outcome. So I think the question is going to be whether or not those reach that clinically effective level or not. And I think it's just unclear at this point. So sorry, I don't have a great readout on when we'll know about those. And then Noel has a question about, do I see these being used in combination with CFTR modulators? These could be used to provide a better lung environment. There is potential that you could use these with CFTR modulators. So I think one of the challenges of an mRNA therapy that I mentioned is you have to get a functional protein. It should work right, but something you deliver doesn't always work totally right. And so it's possible that a CFTR modulator could help your mRNA therapy fold and translocate better. We kind of hope we won't have to, right? Because one therapy would be a lot better. There are, or maybe he's talking about nonsense mutations. And yes, they are trying read-through agents that would make a short protein in combination with modulators. That's something that's definitely actively being pursued and could help boost that function up to the level we need to see clinical efficacy. And I think there's going to be a lot more information on the nonsense and read-through stuff in plenary 1 at NACFC for those who are attending. I haven't seen the slides yet, but I think I heard that that's what it was about. And my apologies if I just gave you bad information. Now that I said it, I'm nervous.
Anybody else have any more questions? We have about 5 minutes left in the hour. Feel free to speak up, raise your hand, speak out, whatever you like.
Thanks, Joan. I think it's plenary 1, but thank you. I do have my story straight. Yep. So yeah, we'll look forward to plenary one and seeing what's happening. This will be my first in-person NACFC, so I'm really excited.
Dr. Germana, I don't know if you mean to have your hand up, but you can close it out if you'd like to.
No, am I unmuted? Yes?
Yep.
Okay, I don't mean to close it out, but I just wanted to say, listening to all of this brings up the idea of how hard we have to fight for our patients sometimes to receive modulators, especially those who respond, when, in vitro, when we send specimens to John Brewington's lab in Cincinnati, it just, as we talk about all these future medications potential for our patients, it just reminds me how important advocacy is, and especially on our state and federal levels. So I think that's an important part of this is, that we continue to pay attention to that as well.
Absolutely.
Okay, well, I thank everybody for joining. I thank Dr. Foshay for taking her part of her evening and providing it to us. And like I said, I will have a recording, which I'll send out, and we'll keep you posted on the Spanish availability. And Joan, I'll keep you in the loop on the slides. So if anybody else needs anything, please don't hesitate to reach out. And thank you again, and have a good night.
Thank you. It's great to be here.